everyone, I'm Vicky Hyejin Yoo, a technical manager for Contact Solutions at CJ Bio. Thank you for joining the CJ Tech Talk. Today, I'd like to introduce COXIELD, a natural anticoccidial. Here are the contents of my presentation. Firstly, I will start with the introduction, and then I will talk about how we verified COXIELD through in vitro and in vivo trials. Finally, I will conclude my presentation. First, I will explain coccidiosis and how we've been controlling it. Coccidiosis is caused by infections with parasite called coccidia or Eimeria. It is a ubiquitous organism and nearly impossible to eliminate from the environment where the poultry are raised. Additionally, Eimeria can rapidly multiply in the intestine of the poultry. So as the parasite proliferates, it destroys intestinal cells, leading to the enteritis and other intestinal diseases. Also, it reduces growth performance in the bird. So this is the life cycle of Eimeria. When a broiler ingests oocyst, shed and feces, the sporozoites are released in the gastrointestinal tract, a process known as excitation. The sporozoites then invade the host epithelial cells. Inside the host cells, the Eimeria reproduces asexually, forming merozoites, second generation or offspring. As a result, the thousands of the second generation merozoites proliferate. These merozoites then develop into oocysts, which are excreted in feces, completing the cycle and spreading the infection. These coccidia broadly spread all over the world and arouse financial losses at $3 billion US per year. So chickens are infected with the multiple species of Eimeria, mainly Arcebrina, Tenella, Maxima, Necatrice, and Brunelli. So each Eimeria strains causes regions in different parts of the gastrointestinal tract, and in severe cases can lead to high mortality. So how can we control coccidia? The coccidiostats are categorized into chemical drugs, ionophore, vaccines, and natural products. Firstly, chemical drugs. They are inexpensive and effective, but develop resistance quickly. And they also require a withdrawal period due to the residue concerns in carcass. Next, ionophores. Chickens develop resistance to ionophores slowly than chemical drugs. Antibiotic effects are weak and cause toxicity if overdosed. For the vaccines, while effective for building natural immunity, vaccines take about 3 weeks, which are almost half of the broiler's life, to establish full immunity through recycling. And administering vaccines also requires a significant labor and time, hence increases production cost. Additionally, the vaccination carries a risk of the initial infection, which can reduce growth performance and cause enteritis. Lastly, the plant-based anticoccidials. These are emerging products as alternatives. They are reported much less frequently from residue and resistance issues, but often shows variation in efficacy, and their mode of actions are not always clearly understood. When you take a closer look at the history of the anticoccidial drugs and their resistance pattern, we can see that no new chemicals or ionophores have been introduced since the mid-90s. The most recently approved synthetic anticoccidial for broiler was launched in the US over 2 decades ago. This graph shows the timeline of when these drugs were introduced. The triangle represents the chemical drugs, while the circle indicated the ionophores. The chart below illustrated the years when the resistance to these drugs was firstly reported worldwide. So as you can see, the resistance developed for chemicals and ionophores shortly after their introduction. So with growing concerns about drug resistance Eimeria and drug residue in broiler meat, there is an urgent need for effective and innovative alternatives to these drugs. So it is crucial to develop strategy to minimize the emergence of resistance in Eimeria and extend the effectiveness of existing drugs. Currently, the several approaches are being used, including rotating multiple products and using different products within a single cycle shuttle program or employing a bio-shuttle program. Whichever strategy you choose, having fresh and effective anticoccidials without resistance issues is essential to minimize economic loss from the coccidiosis. COXIELD is an advanced solution for managing coccidiosis. It combines a powerful blend of phytogenics with a triple mode of actions. So it delivers superior efficacy and delay the development of resistance, and there's no withdrawal period required. Followed by the introduction, I will explain how we verify the product with in vitro tests. As I mentioned earlier, the COXIELD demonstrates efficacy at 3 critical stages of Eimeria life cycle. First, it directly kills sporozoite after excitation. Second, it inhibits the invasion of the host cells by sporozoite. Finally, it suppresses the proliferation of merozoites. By inhibiting these 3 major stages, the COXIELD controls coccidiosis more effectively than other coccidiostats in the market. We compared the efficacy of these 3 MOAs with the comparator's product through in vitro tests. The comparator's product included an ionophore, a chemical drugs, and a phytogenic product. The results confirmed that COXIELD demonstrated superior efficacy at every stage. In contrast, the garlic acid, a well-known natural anticoccidial agent in the market, was inhibited coccidia at only 1 stage. I will provide more details in the following slide. First, we observed MOA1, the ability to kill sporozoites under a microscope. In the control groups, the Etenella cells were alive and intact. When treated with the salinomycin, we saw the head of the sporozoite swell and eventually burst, leading to the cell death. In contrast, with the COXIELD treatment, a whole cell underwent the necrosis and completely killing the sporozoites. However, when treated with the garlic acid, the sporozoites remained alive and shows no changes at all. Second, we examined the MOA2, which involves the inhibition of the host cell adhesion. Let me explain the experiment step by step. First, we prepared the host cells, then we added and spread the sporozoite. Next, we treated the test substance, and after 1 hour, washed the cells to remove any unattached sporozoite. Compared to the control, we found that when COXIELD was applied, the number of the sporozoites attached to the host cells was significantly reduced by up to 60%. Finally, MOA3 focuses on preventing Eimeria from bursting out of the host cells after invading and multiplying. In the control groups, you can see that merozoites has multiplied significantly. The ski joint acts like a breeding sacs filled entirely with the merozoites. However, when treated with the dicrodril, the number of the merozoites were reduced by 53%. And with COXIELD, it was reduced by 66%. On the other hand, the garlic acid allowed the active proliferation of the merozoites. This clearly shows that the COXIELD inhibits Eimeria infection by suppressing the merozoite proliferation. To summarize, COXIELD controls coccidia in 3 ways: by directly killing the sporozoites, inhibiting the host cell adhesion, and suppressing the proliferation. Next, let me explain how the efficacy was demonstrated in the animal trials. The first trial was conducted to measure how much the region in the cecum could be improved and how effectively oocyst discharge could be reduced when feeding diets with the variable coccidiostat to Eimeria challenged broilers. The trial used 360 broilers for 22 days. Regarding the treatment groups, there was an NC group, non-challenged, a PC group, challenge control, and groups treated with the salinomycin and COXIELD at varying concentrations under the challenge conditions. The challenge was performed using the identified field strain of Eimeria. Results show that COXIELD reduced both the region score and oocyst discharge in the broilers challenged with the Etenella. Looking at the graph on the left shows the region score in the SECOM. In the NC groups, no region was observed, and while the PC group shows the significant increased region score. The treatment with salinomycin reduced the region score, while the COXIELD reduced them even further, particularly at the incursion rate of 100 grams per metric ton. The graph on the right shows the oocyst discharge. Before the challenge, no oocysts were detected. However, after the challenge, the oocyst level was increased sharply, and so both salinomycin and COXIELD significantly reduced oocyst discharge with notable effect. And second trial was done to compare the efficacy of COXIELD with chemical agent and other natural anticoccidial products. It was a field test carried out at a research farm in Indonesia using 375 roast broilers for 31 days. The treatment group included NC, PC, and treated groups. For NC groups, dicrodil was treated without a challenge. For the PC group, the dicrodil was treated with the coccidia challenge through an oral dose of the 4 major strains of Eimeria. The other groups were treated under the challenge conditions with COXIELD or 2 commercially available natural anticoccidial products, A and B. Here, the results show that the broilers fed a diet treated with the COXIELD significantly improved the gross performance compared to the broilers fed a diet with the other coccidiostat. Graph on the left shows the body weight. Even with the dicrodil treatment, the Eimeria challenge significantly reduced the body weight. However, the COXIELD group shows the comparable growth rate to the non-challenged group, which is much better result compared to the bird-fed diet with the other natural products. Similarly, looking at the body weight corrected FCR on the right, you can see that the COXIELD significantly improved the FCR than other treatments. Next, we evaluated COXIELD on an eco-friendly farm where the synthetic chemicals are not used. The farm's diagnostic results showed infections with the 3 different Eimeria strains. In this farm, when the COXIELD was added, the region score was significantly reduced from 3 to 0.5. As shown in the photos below, before treatment, the intestinal hemorrhage and severe edema was observed in necropsy. However, after 4 weeks of COXIELD supplementation, the intestine had fully recovered. Additionally, the performance in this trial was greatly improved. The FCR improved from an average of 1.57 before COXIELD treatment to 1.36 after supplementation. In the next trial, we demonstrated that using COXIELD along with the ionophores can be synergistic in terms of FCR and production index. The farm had been using amprolium, but due to the resistance issues, 2 strains of Eimeria were still detected. So we supplemented the COXIELD in parallel. As a result, the combination shows a significant improvement in FCR and performance index, demonstrating that a strong synergistic effect. Here is the usage guide for your reference. We recommend incorporating COXIELD into your program for combined use, but it can also be used as a standalone solution depending on your situation. And please remember, our product is newly launched with a minimal resistance and no withdrawal period. So most importantly, it delivers a clear and proven MOA and efficacy that you won't find in any other natural product in the market. Thank you for listening today, and if you have any questions, please contact CJ Bio representatives in your region.